too many files here. You put your talk taken on the mic. This is more fun. What happens when you're the common computer? Real only. Well, I sent Gina my talk title like a month ago, thinking I was going to give a full science talk, and when I actually set to put it together last week, I reread her email invitation. It said, talk about trajectory, so I'm going to see if I can hit both of these in one presentation. So it's fun to be here. My thesis defense was not in this room, it was Munzer, but I think this is one of the first times getting back here to talk, so it's fun to look at. I think I interacted with, or overlapped with about half of the people in this room during my time here, either earlier or later, so it's great to sort of be part of that larger family. So I'm at the University of Utah School of Medicine in Salt Lake City in the biochemistry department, and we now work on malaria, so I'm going to spend a little bit of time telling you about what our interests are and what we're working on, and then kind of tell you a bit about my trajectory and kind of how I got into enzymology, and then from there into working on malaria. So I pulled up this photo from my PhD defense in 2009, and I got no gray hair in this picture, and Dan has more hair and less gray hair. But so hopefully to weave into this is how training with Dan influenced my trajectory and where I am now. So I thought, like many people here during your time, you collect this library of Dan Herschlag quotes that kind of percolate through your mind as you move forward. And some of these, now as a parent, you realize, well, you think back on your own childhood, and there's quotes that you swear you're never going to say your own students, and then you vaguely start finding yourself using some of these, and it's kind of an odd experience. So I was sitting here trying to think of a few of these, and Jesse's going to share more of these and other tools tonight. And the first few, I'm happy to say, I haven't yet used, but this first one was one of the first ones. I don't remember the context of this at all, what it was, but anyway, that was there. And then there was this one that Dan Kraut and I got when we shared a bench for 4 years, and I'm happy to say it actually was a good idea, so Dan was right on this account. And
that's away from the money.
That's that. And so then the next two, I don't know if current lab members have heard these speeches, but these two actually have found myself giving versions of these talks to my students, which is, you just put in a little more time, this last one, right? So it's, you want to get into log phase of scientific progress as soon as possible, because then it's compound interest at the end. And this is the master one here, for another reiteration of reading and knowing the literature. You're not making use of what's come before you, you're doing them a disservice. And there's some truth in that, which I like a lot. So where are we now, right? So malaria has been eradicated from this country for over 60 years, but this is still one of the most devastating infectious diseases worldwide, with hundreds of millions of annual infections and hundreds of thousands of deaths. Tragically, most of these are in sub-Saharan Africa, largely affecting pregnant women and children under the age of 5 with naive immune systems. And although eradication has been a long-standing goal of the public health community, there have been some real challenges to obtaining this. It's complicated from public health difficulties, the absence of an effective long-lasting vaccine, and a crafty parasite that's really good at evolving drug resistance. So this is a parasite that invades 3 different host environments: mosquitoes inject the sporozoites into the skin that traffic to the human liver, where they invade hepatocytes, grow over 10 to 14 days, at least with the most virulent Plasmodium falciparum. And then that hepatocyte ruptures and starts the asexual blood stage, where these parasites grow with a 48-hour life cycle, replicate, form sort of 12 to 24 daughter parasites that then reinvade the erythrocyte, and then at low levels they stochastically differentiate into sexual gametocytes that are the only form viable in the mosquito that the mosquito takes up in the blood meal when these sexually recombine. So all the clinical symptoms occur in this blood stage infection. This is also the stage that we're best equipped to culture in a laboratory setting. And this ability to do that has really ushered in enormous advances over the last 2 decades in our understanding of the basic biology of this organism. The genome is sequenced, and the early 2000s. Ron Davis's lab here at Stanford played a key role in that. Roughly half of those genes could be functionally assigned. That was great, as I'll talk about, because that raised the prospect of finding new parasite-specific drug targets. But this is a highly divergent eukaryote, and still half of that gene encodes proteins that are so different from well-studied organisms that we really don't have predictive functions for most of those. And so after the genome was sequenced, it was great. People went in, they sort of started annotating these dizzying metabolic maps and said, oh, many of these pathways are really different from what human cells have. There's going to be great therapeutic targets in here. But there's, of course, that's an opportunity. The challenge has been cashing in on this part of the complexity is this is a parasite that's been subject to selective pressures in 3 different host environments. So it doesn't rely on all of these pathways all of the time. You really need to understand what are the critical metabolic functions that it needs. For example, if you want to develop a drug to kill blood-stage malaria, if you want to block transmission in a mosquito, if you want to develop prophylaxis that really gives it this bottleneck of just a few sporozoites getting injected into humans. And so general goals in my lab has been to unravel, what are the critical biochemical pathways, what are the properties of these pathways that underpin parasite survival, with sort of the basic science goal of really understanding metabolic adaptations and the selective pressures that have guided evolution of this pathogen. How are these different from pathways that are encoded in our cells? And then longer term, of course, the goal and hope is that we can leverage this understanding to identify new parasite-specific therapeutic strategies. Now, I like to say, coming into this, this is really a great time for moving into studying an organism that has historically been pessimistically been viewed as intractable to tackle, but has really become much more tractable in the last decade now. So there's really great tools. CRISPR-Cas9 that we've heard about before has had a huge impact. When I started my postdoc, you would just electroporate in a plasmid and drug cycle for 6 months to see if it would integrate to try to tag a gene, and at the end of that, it just didn't go in, so it taken 6 months and you hadn't gotten anywhere. By the end of my postdoc, when Cas9 came online, you could electroporate this in, and in 2 weeks get parasites back, and they would be a uniform polyclonal population of immigrants, and we were off and running, testing something. So there's now great conditional expression systems, especially coming from, a biophysical enzymology background. I've been keen to sort of apply the cellular type tools with cellular, chemical, and physical probes, especially with mass spec-based metabolomics now, genetically encoded biosensors, and now reconstituting systems in vitro to understand molecular properties and build up a higher order understanding of biology. And especially now, there's been a lot of study in the blood stages. Now there's increasing access to mosquito and liver stages and the unique biology that goes on in those settings. So this is a eukaryotic cell. It's got a mitochondrion, but people know. It's also got this really interesting organelle called an apicoplast. It's a relic non-photosynthetic chloroplast. Both of these organelles have genomes, and there's key processes that are targeted to each of these. They're validated drug targets. If you go to a malaria-endemic region right now, you might take a drug called malarone. One of these is an ecovacome. It's a cytochrome B inhibitor. Or you might take doxycycline. It's an antibiotic, which kills prokaryotic bacteria, but it actually is effective against this eukaryote because it inhibits ribosomal translation in this organelle, because those ribosomes are distinct enough from what's cytoplasmic. So these organelles have really stripped down genomes and functions, really fascinating and unusual biology, and there's some really good, because of this simplified biology, there are really good chemical tools to dissect aspects of organellar function that are much harder to do in other cell types. And the goal is, by understanding this, that there might be new druggable targets, especially a large amount of hope in this apicoplast organelle, since human cells lack this. So I'm going to quickly breeze through 3 very brief stories and then talk about trajectory, about how I got into this. So this is a parasite that invades the most heme-rich cell in the human body during blood stage infection. It digests 80% of the hemoglobin in the red cell in 48 hours. Takes the heme from that, sequesters it, and biomineralizes it into hemosome crystals. But heme's not just a toxic byproduct, it's also a eukaryotic cell with mitochondrial cytochromes. It needs heme there, and so it encodes this heme biosynthesis pathway. And there was a lot of confusion early on of why is there a heme biosynthesis pathway if this is living in such a heme-rich environment. And so not a lot was known about that, but sort of part of what we were able to unravel is that you can actually knock out all of these enzymes in the blood stages. It doesn't require a heme synthesis there. It's able to scavenge it, but in the other environments of the mosquito and the liver, then it is reliant on heme biosynthesis. But had we stopped there, that would have been interesting enough, but we took it a step further and realized that some of these developing metabolic probes of heme synthesis activity, that if we put in a precursor, that this was selectively taken out by parasite-infected cells. So erythrocytes during terminal maturation lose a lot of their permeability. They don't take this up at all. When there's a parasite living inside, they're now relying on taking up nutrients from the serum. For example, they don't do amino acid synthesis. They can get 19 of 20 amino acids from human hemoglobin, except isoleucine. There's no isoleucine in human hemoglobin, and so they're relying on taking this up to the medium. So they export hundreds of effector proteins that remodel the erythrocyte, and one of the properties of that is that they enhance the permeability. Turns out we can exploit that. If you put in a heme precursor, it's not taken up by uninfected red cells, down here, uninfected red cells down here, but it's selectively taken up when there's a parasite inside. Red cells in their precursor stages are prolific heme biosynthesizing cells, but they lose that capacity during maturation. But half of the pathway that's cytoplasmic sticks around. Those enzymes stick around in the red blood cell. They're just ordinarily quiescent because there's no substrate that they're getting. Well, it turns out you can actually stimulate that latent activity by putting in heme precursors, because when there's a parasite inside, they'll selectively take that up. They'll synthesize porphyrins from that, which are highly fluorescent. You can see their accumulation out here in the infected red cell cytoplasm. And porphyrins are highly photoreactive. If you shine light on them, it generates reactive oxygen and kills cells. This is actually a form of cancer therapy called photodynamic therapy. So one of the things we've been exploring is taking chemical approaches to activating these porphyrins that selectively accumulate in infected red cells, using chemiluminescent agents like luminol that have great toxicity profiles, good spectral overlap with these porphyrins whose synthesis that accumulate, and their activation is synergistic with the current frontline antimalarial artemisinin. So without going through this, it works really great in vitro. So now we're moving forward to test this in some mouse model systems and explore possible efficacy there. Continued interest in heme metabolism. So the electron transport chain is sort of fundamental to all eukaryotic cells, parasites are no exception. But parasites are fascinating. They've evolved subject to different selective pressures than other well-studied cell types. So we encode a single cytochrome C that sort of moonlights in two roles. One, it's a mobile electron carrier between complexes 3 and 4. The respiratory chain, that's the classical role defined for cytochrome C, and yet it's also known that cytochrome C is a key factor in cellular apoptosis under certain oxidative stress conditions that will now interact with cardiolipin on the inner mitochondrial membrane. It will oxidize it, and that somehow facilitates, and through a novel peroxidase activity, that somehow facilitates cytochrome C release into the cytoplasm that activates a pro-apoptotic signaling cascade. Parasites don't just encode one paralog of cytochrome C, they have two. One that looks a lot like humans, that we have good evidence that really is playing this canonical electron transport chain role. The thing I love about parasites is they have the canonical pathway and they do some funky twist on it. So they actually have a paralog, a gene duplication in a paralog that's the most divergent cytochrome C that's known for any eukaryotic cell. And so we've been trying to explore what are some of the functional properties of this. It's really divergent, just N-terminal to the axiomethionine ligand, and from the structure up here coordinates that iron. That's known to strongly influence the reduction potential of that heme. And so if you just take UV-vis spectrum with bound heme in the presence of a strong reductant, the canonical cytochrome C that the parasite encodes is very similar to the human enzyme. So these sharp peaks here are expected for a reduced ferrous iron bound to cytochrome C, but this really divergent one has these amorphous peaks here that show that it's almost certainly still oxidized and has a much smaller reduction potential. So I don't have time to go into it, but we think there are some interesting implications for where these cytochromes are differentially important in the parasite's life cycle. This canonical cytochrome C as a canonical electron transport carrier, but this really divergent copy that we have data that this is really not required in the blood stages, but early hints that this is playing a critical role in the mosquito and liver stage, which is actually where mitochondrial function really gets upregulated. We think there's a role for this as a constitutive peroxidase in that environment. Got one last story I'm actually going to skip through because I'm probably getting a bit short on time here. I actually gave a talk here yesterday to the parasitology community, and a few of the people from Dan's lab showed up to that, where just the last example of a thing that looked like a hemoxygenase. It has a hemoxygenase fold, but it's actually lacking degrading activity. and strikingly, now that we have a knockdown of it in parasites, seem to play a strong role in regulating, transcribing the small genome in this apical-plast organelle that we're just starting to understand, and having some fun time with that. All right, so how did I get from studying enzyme function in Dan's lab into studying malaria? So as an undergraduate at UCSD, I worked with Susan Taylor. I knew I had strong interest in bioorganic mechanism, and towards the end of my time as an undergraduate, took a senior enzymology course, and actually at that course read Shuo's paper, PNAS paper that had come out, which I understand from the lore was her rotation project, I think, is the data from that. And I thought this was just really fascinating that you could take a small molecule model system and learn something deep and interesting about enzyme function, that you could study something in a systematic fashion and really isolate properties from there. And so that kind of put things on the roadmap to me of thinking longer term for the types of science and approaches that I wanted to do. I didn't go straight to graduate school from my undergraduate days, and this is maybe where the early thinking of merging mechanism and malaria came together. Is I actually spent 2.5 years as a Peace Corps volunteer in Ghana in West Africa, teaching high school chemistry here in the far western region. This is a malaria endemic region, so I had access to prophylaxis and mosquito repellent and bed nets, and really didn't deal with malaria at all myself, but certainly had students who didn't have access to this, even though there was widespread chloroquine resistance. If they did get sick and they did go to the hospital, they still got a shot of chloroquine in the butt and sent them on their way, which was not doing great things for helping out with chloroquine resistance. So I had a lot of time here to think about what I wanted to do long term, the type of questions I wanted to go after. So came back from that, applied broadly to graduate school. Susan's advice was, go to Berkeley, there's great people there, that's who she knew. Mike Marletto was there and tried hard to recruit me. He actually had a technician working for him at the time that was a Peace Corps volunteer with me in Ghana, so that was an interesting connection. But opted not to go from that, came down here to the farm, and Mike still gives me crap for coming to Stanford and Berkeley, but that's okay. I actually met Jesse on every graduate interview except Stanford. I ended up coming here, joining the same lab, working on the same half of the lab, and even in the same day, and ultimately picked Stanford because of the culture of the biochemistry department here, because I wanted to study enzyme mechanism, because going back to Shuo's work there and what that represented, nobody else was really doing the kind of work that interested me as much as Dan's, and so it was actually a pretty easy decision there. So I dug up this flyer from my defense, and I think all of us commented on the rigorous training we get from Dan, thinking critically, reading and knowing the literature, thinking deeply, and I think the first part of this came from someone else, and you added the second part.
It came from somebody who I heard recently met in your stall.
Oh, all right. Well, I'll mention Neil again in a second, right? So think deeply and extract as much as you can from the data there, but obviously you need some amount of data to make it real. But what I really like here is really be intellectually invested in the work of others. That's be a good colleague. Now as a faculty member, obviously I'm building my lab now, but I like thinking about other people's science. I like giving them feedback, and I'm really trying to encourage people in my lab to, don't just view what you're going to get out of it, but for yourself and giving feedback to others would be a good colleague. I like this one as well. Don't be jealous of other people's success. Be inspired by it to bring out your best work and move forward, and be generous in giving credit to others as well. Of course, we had some fun times in the lab here. Jonathan and I did a fun climb to Mount Ritter together during my time here in the Eastern Sierras, and then I think this is still on Dan's website, in fact. We actually did a traverse from Fallen Leaf Lake across Desolation up and over Pyramid Peak there, and this is from the summit of Pyramid Peak, which was fun. Dan also had good friends and was very supportive. I was thinking about what I wanted to move on to at the end of my time. So many of you know Pradeep, who was here and who passed through and had really good talks with Pradeep as I was thinking about what I wanted to move on to next. As it turns out, I discovered I'm actually related to Pradeep. Pradeep did a first postdoc. He knows Dan from his second postdoc with Bob Abels and Brandeis, but before that, he actually did a first postdoc at the University of New Mexico. I discovered this when I was sort of digging at paper as well, with a guy named Phil Reyes. Well, Phil Reyes is a great uncle on my dad's side. He's the only other person on either side of my family who ever did a PhD, and I think it's just crazy coincidence that Pradeep was a postdoc with Phil. I actually met Phil for the first time when I was moving out to St. Louis to start my postdoc. So people have talked about these random coincidences in science that come up, and it's really bizarre how these things work out. Pradeep and I see each other a lot at meetings now. So this is at the Parasite Gordon conference this last June out at Rhode Island. So here's Pradeep, here's me, and then Pradeep said, oh, well, look, here's Neil Stahl right here at this meeting. It turns out Neil's married to Jane Raper, who's a parasitologist in the field, and Neil comes to all of this, and so I had a fun chat with Neil, and certainly cited Neil's paper many times during my early work there. So again, it's these crazy connections that bring people together from disparate fields. The last one is when I left Dan's lab, and I knew I wanted to move into parasitology and study mechanism. I went out to WashU and did a postdoc in microbiology, which of course is the headwaters of Stanford biochemistry, where Arthur Kornberg was when he sort of gutted the department there in Flyover West. And you still haven't forgiven him for that, but it was interesting to retrace those steps, and this is where I sort of had to pick up actually being a biologist and learn all sorts of stuff that I didn't know before. So just wrapping up here, of course, you feel like when you've been the benefit of great mentorship and people that have come before you, feel a strong desire to pass that on to the next generation. So, you know, there's a lot of hard things about starting a lab. People told me the personnel side would be among them, and it's certainly true. And so trying to figure out how to be a good mentor to graduate students, how to inspire them, how to pass on all of these lessons that we've learned along the way from great mentors. But that's part of the fun. So thanks, Dan, and look forward to continued support and interaction.
Hey, we have time for one question. Good.
Dan, go ahead.
You can each ask half a question.
Have you tried? No, you couldn't.
Oh, so it's a haploid genome.
Yeah.
Oh, wait, was he asking a question? Well, I'm asking about a half a genome. So it's a haploid genome. It seems like it makes functional screen should be
Easier. The challenge was that genome editing. So part of it, so there's 5 species that infect humans. Falciparum is the most virulent. It also has the most AT-rich genome of any organism, greater than 83% A's and T's. So it's molecular biology pretty tough. And again, prior to CRISPR and editing efforts, you know, it could be pretty tough and time-consuming to just tag genes for localization studies. There weren't good knockdown tools, so studying function was hard. And so that's all kind of come together now. We have great genome editing tools. We actually have some robust gene knockdown tools. And so combining that with functional studies in cell and in vitros.
Okay. Felix, question?
Well, the second half of my question is to knock out cytochrome C2. So what's the phenotype?
So there's no phenotype of the blood stages.
Okay, so
Yeah,
and
and
that's
so
the reason why
our goal is now to passage that into mosquito and see what happens. So the transcript, the mRNA transcripts of that are massively upregulated in the late mosquito stage. That's where we predict it's playing a function.
Okay.
All right.
Thank you.